potential for the future. Let me start with the words from Dr. William Foch. Vaccines are the tugboats of preventive health. We all are well aware that vaccine development is extremely challenging, complex, expensive, and a high risk endeavor. But there is no doubt that vaccination is one of the most cost effective health interventions available, saving millions of people from illness, disability, and death each year. Thanks to vaccination, smallpox is now a thing of the past, and we are on the verge of eradicating polio. New vaccine technologies able to achieve rapid development as well as large scale production are extremely important. In the last 18 months, we have witnessed some of the most remarkable scientific breakthroughs in creating and delivering life saving vaccines in never seen before record time. Today, we have the privilege of having experts, policymakers, and the industry leaders in the field of vaccinology on a single platform to discuss and exchange ideas and the lessons from their efforts to develop and manufacture effective DNA based vaccines towards the global fight against COVID 19 and extend their knowledge to also help address other diseases. With this, on behalf of Department of Biotechnology and BIRAC, we would like to welcome all to today's symposium and would now invite Dr. Alka Sharma, Scientist HDBT and Managing Director BIRAC, for her opening remarks and introduction to the symposium. Over to you, ma'am.
Thank you, Shikha. Thank you for the brief introduction. Good evening to our Indian participants and good morning to our US participants. On behalf of Department of Biotechnology and its PSU BIRAC, I welcome each one of you for today's International Symposium on Addressing the Pandemic with DNA Vaccine Potential for the Future. Today we have with us Secretary, Department of Biotechnology, Dr. Renu Sarup, and the Chairperson of BIRAC. We also have Dr. Vinod Paul, Member Niti Ayog, Dr. K. Vijayaraghavan, Principal Scientific Advisor to the Government of India, Dr. V.G. Somani, Drug Controller General of India, Dr. N.K. Arora from England, Dr. V. Ravi, Dr. Pankaj Patel with his team from Jadis Cadilla, Dr. Steve Reed, Dr. Vadarajan from IIC Bangalore, and Dr. Hangli from CEPI. As we all know, the vaccine are one of the most cost-effective health interventions for combating infectious diseases. The classic vaccine platforms, which have been licensed for a majority of diseases, include live attenuated or inactivated whole virus or protein subunit vaccines. However, classic vaccine platforms may not be suitable for all pathogens, especially those that require to be grown in BSL-3 conditions. So in this case, next generation vaccine platforms such as nucleic acid vaccines or viral vector vaccines gain importance, and this has been demonstrated in COVID-19 pandemic. India, which has identified vaccine development as key strategy to combat COVID-19 pandemic, is also developing vaccine candidates belonging to both classical as well as next generation platforms. So you all are aware that of the 6 COVID vaccines that has received emergency authorization in India, 2 vaccines, that is inactivated vaccine Covaxin and DNA vaccine that is developed by Jadis Cadilla, have been developed indigenously. The DNA vaccine ZyCoV-D, developed by Jadis Cadilla with extensive support by Department of Biotechnology and its PSU BIRAC, is the world's first DNA vaccine for COVID-19 that has received emergency use authorization. The development of these vaccine candidates was made possible through the strengthening of a robust end-to-end vaccine development and manufacturing ecosystem across the country. Over the past 3 decades, the Department of Biotechnology has made concerted efforts to strengthen the Indian vaccine research and development ecosystem through active international partnership and implementation of innovative funding mechanism. The most significant global partnership include Indo-US Vaccine Action Program, that is VAP, and CEPI mission in partnership with Global CEPI, partnership with European Union through Horizon 2020, and also partnership with Melinda Gates Foundation. Innovative funding mechanisms are being facilitated through the National Biopharma Mission, and as you know that this is an industry academia collaborating mission and which is being implemented by BIRAC. These efforts have resulted in the strengthening of the ecosystem for vaccine development in the country. Capacities and infrastructure for preclinical and clinical vaccine development, including facilities for animal challenge studies, immunoassay and clinical trial sites, have been set up. Enabling regulatory guidelines have also been issued by the Government of India. This symposium attempts to highlight the efforts of Government of India in fostering vaccine development and the regulatory scenario for vaccines with a focus on DNA vaccines. Also, there will be a session on challenges in the development of DNA vaccines, importance of DNA vaccines for COVID-19, and beyond and emerging technologies in vaccine research. So I hope that symposium will provide valuable insight into the utility of DNA vaccine as a potential technology in overcoming future pandemic. So with this now, I invite Shikha to take us to the agenda for today's international symposium. Over to Shikha. Thank you.
Thank you, ma'am, for the overview. Thank you so much. So, with this, I would like to invite our first speaker. It is our privilege to have respected Professor Vijay Raghavan, Principal Scientific Advisor to the Government of India, joining today's discussion. Sir's office aims to provide pragmatic and objective advice to the Prime Minister and Cabinet on matters related to science, technology, and innovation in partnership with government departments, institutions, and industry. With that, I would request Sir to share his views on vaccine past and present. Over to you, Sir.
Can't hear you, sir.
Sorry, my audio was off for a moment. So, is it to me? I'm supposed to start?
Yes, sir. Yes, sir.
Yes.
Okay, thank you very much. So this is a great pleasure to be at this meeting, and I'm sorry I had some technical issues getting my audio and video on. I heard most of what Alka had to say, and it's really wonderful to have all of us here today. Uh, Ravi, Dr. Arora, Dr. Vinod Paul, Dr. Pankaj Patel and his team, Dr. Renu Swarup, of course, and everyone else from BIORAC and from the Department of Biotechnology. I will not comment substantially on DNA vaccines, but let me be rather brief and talk a little bit about vaccines in general. And this is now a time going forward for us to make a critical evaluation of where we have come from and where we need to go, and importantly, how we need to go. Well, let's look at first where we came from. We came from a situation where India was recognized as a vaccine producing and exporting nation in bulk for a particular set of vaccines, which we were a big player because of volume. In other words, this was like us being a big player in terms of generic drugs. And our vaccine companies, Zydus Cadila amongst them, Serum Institute, Bharat Biotech, Panacea, and others, really rode on this capability, which is not to be sniffed at, very important capability of being able to take vaccines into scale, production, quality, and export. Now, what did the pandemic do? The pandemic brought a sense of urgency, which brought together our, small academic research laboratories in the vaccine field, startups, medium-sized companies, large companies, to not only scale up what was feasible, and you saw that in some examples, I'll name them, but also start developing afresh. In terms of scaling up what was feasible, I would put in the Oxford AstraZeneca vaccine scaled up by Serum into that category. And there, while you can argue that this is a imported vaccine, Serum's extraordinary capability to manufacture in quality and to scale must be appreciated. And as a result of that, you have the bulk of the vaccination today being rolled out by Serum. So, this brings to light the critical point that, you can design something, you can get it through the early stages, but scaling up relentlessly is a capability which we must also develop and broaden. You cannot have a situation where this is the hallmark of one company. Multiple other companies need to come up. So one shouldn't sniff at saying that, oh, this is an imported vaccine or a vaccine which is not developed here. That's, of course, important, but the scaling must be appreciated. Now, next, we must appreciate the extraordinary speed with which the partnership between ICMR and Bharat Biotech resulted in the development of an inactivated virus vaccine. Now, again, that some would argue, those who are looking at new technologies, that this is not rocket science, new science, but to do this properly, to inactivate properly to scale, and to go through your steps of ensuring, safety and immunogenicity is not a small matter. And again, ICMR and Bharat Biotech and the Department of Biotechnology, which partnered in various trial sites, need to be congratulated. So that's not a small matter. We would have liked to see scale coming in over here. And if you want to point out what the issues are, the speed of which all the trials were done, and the documentation and the scale, these are areas where we need to go up substantially regarding this. Now, there are other vaccines which are in the pipeline or in late stage of development in a combination of partnerships. Zydus Cadillus DNA vaccine, which I'm sure others will talk about, is one. And kudos to them for taking this on, because DNA vaccines, so far, if you would ask a vaccinologist to say DNA vaccines, what are the chances they'll work? The short answer will be better not to try that. There have been many examples where DNA vaccines have been tried, but they don't, for a variety of reasons, which all of us know, they're not easy to take forward. So therefore, great congratulations to Zydus and his team for taking this to the stage. It's very complicated. It has extraordinary benefits in multiple contexts, as we are seeing and which we will see. And so that's a very good aspect. Similarly, congratulations to Genova and the mRNA team for taking that on. I know there are other companies which have taken on multiple components of the ORF and have started developing vaccines. The team, we have representatives here, Raghavan Varadarajan and others who have looked at vaccine development, which are like at room temperature. They have shown a really good example of how the academia can partner with startups to develop that. There's a great room now for that to develop and scale up. Two other points. One, as we've heard from Alka, the Department of Biotechnology needs to be congratulated on developing sites at THSTI and multiple other places where companies can come in and get their work done. That's very, very good. At the same time, we need to take a self-critical look at how our research institutions, the National Institute of Immunology and the Translational Health Science and Technology Institute in particular, but also other institutes of the Department of Biotechnology, whether they as an institution, as distinct from BIRAC, as distinct from the THSTI's application end, how much they have moved in this situation in vaccine development. Perhaps there's a room for much ramping up on that front. Other institutions also have done well in developing testing capabilities, but clearly we need to ramp up our academia vaccine platform capabilities so people can pivot and go into vaccine development in a big way. The kinds of difficulties in those in academia have had to face in going into vaccine development has been substantial, and that needs to be reduced substantially going forward by creating more interface technology access at multiple kinds of institutions. It doesn't have to be in just one location. This requires resources, but not that much resources, and that can be done on a substantial scale. Let me end by saying that there is another aspect which we need to go forward in a very important manner, and that relates to our vaccine industry now taking advantage. Why is Renu's label VG Somani?
I was wondering that, sir. I was wondering how I'd become Somani, but that's all right. You continue. They'll change it from the back end.
Okay. So going forward, not only was we develop vaccine platforms around our research institutions so people can easily go and start developing vaccine at early stages. But ease of animal research must go very, very rapidly, whether it's DNA vaccines or mRNA vaccine. Toxicity studies and efficacy studies need to be ramped up, and it's about time we just grasp those nettles really squarely. Enough is enough. That's something which should not have people running from pillar to post. That's a very important aspect which we must do. It started now. You must keep up that tempo. Finally, there's a question of resources. Risk mitigation is critically important. Unless risk is mitigated, companies will have a tough time in developing vaccines against diseases which are not yet endemic or a threat or pandemic. If they have to develop vaccines in an adventurous manner, then risk should be mitigated. And that risk should be mitigated, can be mitigated by a partnership with our research institutions and companies, small, medium, and large on scale. You companies such as BIRAC and the DBT have shown the way, but that is way has been shown as a single kind of example and has resulted in a substantial expansion from zero to what we have today. So that's remarkable. It's extraordinary going from zero to something. It also is always infinitely better than what it was. But going from something to double is a very big challenge, and that's the challenge which we need to take up now. So thank you very, very much, and kudos to the Department of Biotechnology and everyone else here for all the work they've done. And I'm sure there'll be much more happening very soon. Thank you very much.
Thank you very much, sir, for your kind words. Now I would like to invite our next speaker. Um, as we all know, vaccines undergo a rigorous review of laboratory and clinical data to ensure the safety, efficacy, purity, and potency of these products. I would like to invite Professor Arora, Executive Director, England Trust International, and Member Chairperson, COVID-19 Working Group in TAGI, to share the reviewer's perspective on the pathway of DNA vaccine development. Over
Thank you very much. Yeah, thank you very much. Uh, I completely echo what Professor Vijay Raghavan was saying, that from a vaccine manufacturing country, we are transitioning to vaccine developing and manufacturing, and the way scaling up of manufacturing capacities have taken place. It is a kind of, I would say, amazing as well as awesome. Regarding I'm kind of replacing Dr. Somani because I think he has not been able to come. But the process we are finding, we are part of Antagi, but the process, the way it has been, that uh, the country modified the regulatory environment and regulatory processes right in the early part of last year, 2020, April, May. The revised regulatory processing issues started. And one highlight was that issue of emergency use authorization came into existence in this country. The second important point has been that there is a rolling process of evaluation. And this rolling process was taking place at least at two levels. One is at the Antagi level. Antagi, our committee, the COVID-19 working group, has been reviewing the data from preclinical to clinical and then post-regulatory approvals. This was in addition to what the funding agencies have been doing, that is DBT or any other funding agency. And along with this, the data is also reviewed by subject expert committee of DCGI, because only after their recommendation, the the DCGI gives approval, and before going to the program, again, NTAGI closely reviews all the data, the trial data, the immunological data, the animal data also, as and when an additional analysis if required. So, giving example of DNA vaccine, ZyCoV-D, this has been a hallmark in a sense that last year when their application first came, there was a skepticism because previous attempts at DNA vaccine had not succeeded. And Ravi is very much there. He was part of that group, and there was a lot of concern that nothing may come out. However, I think DVD decided for this high risk investment, but such a pleasant investment that today we have world's first DNA vaccine, which is approved. The second thing is that after the subject expert committee reviewed the data prior to approval, and then DCGI gave them the approval for rollout, the issue came to our working group, COVID-19. And here I would say that broadly, the data is very convincing. It is safe vaccine. It is immunogenic vaccine, but the issue got stuck because for children, the immunogenicity data was very limited because we were given to understand that children were taken up for the trial in the later part. So post second dose and third dose immunogenicity data was still not available. So the current status is that vaccine has been approved for rollout in adults, but for children we are waiting uh, for the additional immunogenicity data. The second dimension was the whole concern of several technical experts was the issue of integration or the safety of the vaccine and integration into the host genome or the beneficiary genome. And for that, the SEC had taken great care in a sense that the literature was looked at, as I'm given to understand, because we invited one of the SEC members for our own meeting. We also realized that the company had used the most recent WHO and FDA guideline for developing DNA vaccines. The next generation vector was used for developing the plasmid DNA plasmid for this particular vaccine. So it was very reassuring that the the manufacturer had taken all the due diligence in terms of following the international guidelines, and the data was presented. We were also informed that animal studies have been done, and within the animal studies, the safety and immunogenicity was excellent. So the, the the process I am trying to communicate is that whether the regulatory processes have been made much, I would say, client friendly to or a product friendly. One. The second is there is a rolling process of reviewing the data, and the manufacturers are given opportunities for coming back with modified data or reanalysis. And then there is another layer of safety, like COVID-19 working group of NTAGI, which relooks and kind of reconfirms. So the whole process in the country has been prepared in a way that safety and efficacy and effectiveness is not compromised in any way. And I think this is a paradigm shift as compared to the previous years when bridging trials were done. You look at the immunogenicity and then allow for marketing authorization. But the responsibility on the country increases. Not only that, because we are vaccine manufacturing country now, that the same vaccine we think is going to be used globally, and therefore all due diligence is required within the country before the applications go in either for WHO emergency use listing or other countries. I will stop at that and welcome if there is any clarification.
Thank you, sir. Definitely, it is the fast review with no compromise on the safety and efficacy and the quality of the product that we have the first DNA vaccine for COVID 19. Thank you for your words, sir. I would now like to invite Dr. Amy Swaroop Ma'am, Secretary DBT and DST and Chairperson BIRAC, for her remarks and sharing Indian efforts on vaccine development. Over to you, ma'am.
Thank you very much, Shikha, and very good morning, good afternoon, and good evening to everyone who's joined from different parts of the world. It's a pleasure to be here today, and thank you, Professor Vijayaraghavan, sir. I was trying to look out for your name there. Thank you so much for agreeing to be here, Dr. Arora, and all the other experts, and most importantly, I think the Zydus team, led by Mr. Pankaj Patel, Sril Patel, all of them who have brought out the world's first DNA vaccine, which gave us this opportunity to host this webinar today to have an international discussion on this. We are very proud of this achievement. We're very proud of the achievement which has been done by our Indian company, but we are very proud of being able to have this whole ecosystem for vaccine development and the response that our ecosystem gave to this COVID-19 challenge. Professor Vijayaraghavan correctly said that from vaccine manufacturers, the recognition to being vaccine developers, key cutting edge technology research, I think that itself is something that we are so very proud of. COVID did pose a very huge challenge for all of us. And this challenge was not just for the S&T solutions to be brought in, but handling COVID, working amidst all the various, the challenges of the situation, whether it was the lockdowns, whether it was the supply chain disruptions. It gave us one advantage, and that was the advantage of collaboration. Enhanced collaboration, enhanced connectivity, which, thanks to the virtual platform, everyone connected with. India was in a unique position where we started our discussions for the whole R&D roadmap very early on. And we were, in fact, one of the first countries which presented its R&D map to the WHO Blueprint Group in February 2020. And if I recall correctly, the Zydus Cadila team will correct me if I'm wrong. I think it was somewhere around that very time that we had a discussion, the first discussion with the Zydus team to take on this very, at that point of time, a highly risky, innovative technology that was to have a DNA vaccine for COVID-19. Considering the fact that the DNA vaccines which are in development globally were all either for veterinary vaccines and we didn't really have a human vaccine. I think it was the confidence that the team had on themselves that allowed DBT and BIRAC to put in that level of confidence in them to say, yes, we are ready to work with you as partners to go ahead. We're glad we did that because this is where we stand today with the world's first DNA vaccine. This has been a long journey of connecting different stakeholders, a long journey of building a very robust ecosystem, and as Dr. Arora rightly said, of a stringent regulatory ecosystem for which every vaccine that has come out through this ecosystem has actually passed the test in terms of the highest level of standards that it could be. But this whole journey has not been very simple. It's been a journey full of challenges. And the researchers here, and I'm not going to stand between you and the Zydus team and others, because they're going to share with you the actual challenge that they faced. And I think eventually what is important for us to see is today we are not talking just about a DNA vaccine for COVID-19. What is the importance of this platform? What is important for us to say is what is next that we're going to do. Yes, our immediate priority is How do we get the DNA vaccine into the immunization program? How do we get large scale manufacturing of this vaccine? How do we get it to the entire population it's meant for, whether it's for children or for adults, depending on the approvals that it has got? I know that Zydus team is looking at different trials, whether it's a two-dose trial or others, but I think what's important is how do we use the knowledge that we've generated with this uh, DNA vaccine to be able to be used for other such diseases. We know that it's an important platform. We know that it's gone through a rigorous test of all best scientific procedures and processes. And with this understanding and with this information, what next can we do? Today, the country has one of, as I said, a very robust ecosystem, whether it's our animal challenge facilities, whether, and I think all the members here will recall that when we began, it was that time we were still looking at whether we should do the, where should we take up the non-human primate studies, where should we take up the animal challenge studies, would we have laboratories for immunoassay studies enough for all the vaccines that are coming up, will we have enough clinical trial sites to take up these various clinical trial sites. Today, for each one of them, we have a response. We have the non-human primate facilities available. We have the animal challenge facilities. We have the immunogenicity laboratories recognized by the CEPI network. We have, and I know that the National Biopharma Mission, which is set up, has set up facilities, and the Zydus Cadila team themselves have been partnering with these facilities to give us the best of results. But having said all this, being sort of uh, pleased with our success over this last 18 months in terms of what ecosystem we've been able to build through DBT, through BIRAC, through working with Health Ministry, working with the Pharma Ministry, working with ICMR, the regulator, everyone. But I think now it's important for us to say, what do we do next? How do we take this into the next level of not only the technology, but also looking at the diseases that we'll be able to target? I think the Zydus Cadila team has very good understanding and knowledge of it. I'm hoping that in the panel today, we will have discussions which will throw light on what really could be the next steps. But I would like to conclude by saying we're always looking at excellent challenges to be posed before us. And the team here knows that for the last many months, we've been discussing on how do we really from India take on a pan-coronavirus vaccine. This is clearly a platform that we should look at it. We've had discussions. We've probably, with our discussion with CEPI, with NIH, with others, have given us sufficient understanding on how we should move ahead. But yes, this is clearly a platform that you may want to consider taking into the next stage now. And I would hope that the experts today will give us sufficient understanding because India is ready now to take on that next new challenge for vaccine development. And this is not going to be just for our own diseases that we are looking at, for many of them which we are working in vaccine development, but even for global challenges. So thank you all for being here today. This is a webinar that we're going to discuss some important aspects, but also prepare for ourselves a roadmap for the future. So thank you so much for having me here, and over to you, Shikha, once again.
Thank you, ma'am. Thank you so much for the inspiring thoughts. And we do look forward to more challenges and more successes in the future. SARS-CoV-2, as we all know, is the DNA vaccine, DNA-based vaccine for COVID-19 developed by Cadila Healthcare. I would now like to invite our next speaker, Dr. Pankaj Patel, Chairman, Cadila Healthcare, to share his experience and challenges of developing a DNA vaccine. Over to you, Dr. Patel.
So, good evening and good morning to all the eminent panelists. And we from Zydus team, Sarvel Patel and my colleague Kapil join you in presenting our current achievements on the vaccine journey and also a little bit about how we began this journey for our company. It's been, while successes are always praised, I mean, we have gone through a lot of challenges as well. We had two vaccines implement, one with the measles vector-based vaccine, and the other one was the DNA platform. And we are very happy that we have been successful in bringing one of the vaccines to the emergency use authorization. If I can, for the company, just a brief background. in this business of pharmaceuticals and life sciences for the last 70 years. And what is very important and heartening for us is that, today we are definitely one of the largest integrated biologics and vaccines portfolio companies in India. So it's from a period of last 20 to 25 years, we've been putting in a lot of effort on the research to make sure that we have effective capabilities in developing and being self-sufficient when it comes to India. Kapil, can you go to the next slide, please? And as you can see, we have a lot of research centers all the way from small molecule to large molecules. And we are today consistently a company that spends almost 8% of our R&D revenues on R&D. And this is the commitment that we have made to ourselves and to the scientists to make sure that we continue to innovate when we talk about the research efforts at Zydus. Next slide, please. Also, I think what is also important is we are just not a vaccine developer, but we have also tremendous amount of R&D and clinical capabilities when it comes to doing research. And two of our programs are now in the global programs where we have trials going on in the United States and Europe and other countries, which are global trials for, again, one of the unmet medical needs in the areas of NASH and PBC. And we have orphan drug and priority review status for these developments for saroglitazar and also for desidustat, which will again be the world's first in a way of bringing an oral compound for anemia. So the history and the success of the company has been that all the way from developing small molecules or large molecules to doing the preclinical, the toxicology work, and the clinical work, all of this is done in-house by the company. And we have been made self-sufficient in terms of being able to do this and also capability of filing these doses in the international regulated markets of the US and Europe as well. And these are the successes that we hope will build a long way for us in terms of the drug discovery pipeline that we're trying to build. We were also one of the first companies in India to also launch the Vaccine Flu, which is the H1N1 vaccine in India, and now the quadrivalent flu vaccine also. We had the first generic of the world's first biosimilar. We have launched antibody drug conjugate molecule, the first generic in the world to be launched in the country. And finally, also we have partnerships with the WHO to launch a dual monoclonal therapy, which is again the first in the world to be done so. And now we have obviously the ZyCoV-D vaccine, which we will speak to you about. This vaccine has been a lot of effort in the making. It's one of the largest trials that has been conducted in India, where we are now about more than 30,000 volunteers who have been administered this vaccine. And we are very happy with the science, the safety, and the efficacy data that we've been able to see for the vaccine. And I'm sure Kapil will speak to you about that. And also the safety parameters and the markers that were very essential to make sure that we succeeded. We as a company did promise that we will combat the virus in all fronts. So whether it was preventive, whether it was diagnostics, therapeutics, or prophylaxis, Zydus has played an important role in not only in India but internationally making many of the drugs available for the global fight against COVID. Next couple. These are just a few, but we have a large set of products that we are developing. I'm also very excited to say that not only in terms of detection kits we have been working on, and we are partnered with DBT and others to make sure that we can bring these products to market and ICMR, but also one of the only Indian companies now to have a cocktail of monoclonal antibodies as a therapeutic, which is under clinical trial now in India. So this we hope that once we're successful, this will be a monoclonal cocktail therapy which will have a longer half-life than the current modes of treatment that are there in this area, and a far more better safety profile with removing the issues with ADCC effects. And we believe that this could potentially also be an important pathway in the forward fight for COVID, when we need effective therapeutic solutions for people who need treatment and who hit severity or who are comorbid. So we are very excited that we work on all fronts, whether it is small molecule front, whether it is our vaccinology front and the large amount of vaccines that we produce, and the monoclonal therapy side, where we have the capabilities to do this internally as well. Next couple. We do have a large portfolio of vaccines. We have more than 20 years of experience now in vaccinology, and we have capabilities on all types of platforms. And we are very excited with this DNA platform. As a company, we are very excited that not only did we develop and we became successful on this side, but We build large-scale manufacturing facilities, one of the largest in Asia, and some of the capabilities on the downstream are probably the largest in the world now in terms of downstream purification. We also have also mixed in terms of bringing the applicators and the way to administer the vaccine. We have completely integrated it, and we are producing many of these components in India now, so that we can effectively roll this out on a larger scale, which is going to be the need of the. And with the data that we have seen on safety, we are very excited that this vaccine can has a multiple purpose use, not only for the current vaccination, but as booster doses or as multiple variants become more critical, how do we build a multivariant vaccine? And the efforts on that are also ongoing. So I think it's a journey that has just begun for the company. Obviously, we have a long way to go. And I believe the evolution of science that is happening and the understanding of COVID-19, we believe that these vaccines will become more part of the immunization programs globally, and probably a yearly cycle or more. And multivariant strategy could be a great opportunity, and that's where recombinant vaccines will offer an opportunity to build that capability. We also work on other technologies like mRNA and others, and we believe in the future we will have technologies beyond just the one of the recombinant technologies, but both, whether it's the DNA or mRNA, we will have both capabilities in the times to come. I'll hand over to Kapil to really talk about the current vaccine and where we are on this journey. Thank you.
Thank you, sir. And good evening to all of you. I'll just quickly take through a discussion on a ZyCoV-D and specifically on the topic which was assigned to us on the challenges we faced during the development of the plasmid DNA vaccine. As Dr. Swaroop mentioned, I think it was somewhere in January of 2020 when I had met her and Dr. Alka Sharma in their office in Delhi, and they encouraged us to move ahead with this concept. And I think we came a long way from that first meeting. And during the course of this development, there had been a lot of support. I remember the first meeting we had where Professor Vijay Raghavan, and Dr. V.K. Paul chaired. And in fact, they shared their personal numbers to, in case any challenges we face during the course of the development of the vaccine. And subsequently, there has been so much of support from regulatory bodies. Dr. Somani has been very kind in advising us, and now Dr. Arora has been being tough, at the same time advising us what kind of data should be available to take this forward for global use. So having said that, I think the merits of the DNA vaccine platform, I think, is quite evident in terms of safety, as it is non-infectious, does not have any risk of anti-vector immunity. So in case of waning antibody response or change in strain, a booster dose of this vaccine can be easily given. It does not include any adjuvant, in our case also no HCPN. So there is no risk of added adverse reaction because of the formulation. And it's classically known that DNA vaccines stimulate both the humoral and cellular arms of adaptive immune response. And that's what we also found in our vaccine candidate. It is also highly adaptable. One can make new constructs very rapidly, as one would have expected, and now we are seeing that new mutants are emerging for SARS-CoV-2. The product is very stable, and manufacturing requires a BSL-1 level of facilities so that it can be easily tech transferred and multiple facilities could be used in case we need to scale it up. Just comparing the different platforms, you would see that a higher dose of administration is possible with the DNA vaccine. One saw a lot of adverse reactions in the initial clinical trials done by Moderna and Pfizer at a higher dose. Similarly, a booster dose, as I mentioned, can be given with this. It's a better safety profile, no adjuvant or excipient requirement, and the cold chain requirements are minimal. And I'll talk a bit about each one of them as I move forward. So currently, this is the global scenario of the DNA vaccines which are in development. As you will see, 12 of them are in clinical development. Two of them are the Zycov D candidates, one having received emergency use authorization. We are also in phase 3 clinical trials for a two-dose regimen, which is underway right now. And interestingly, you see the different kind of delivery devices which are used. Inovio uses a Selectra system, which is an electroporation device. Same thing being used by Genexin. Then there is a PyroDrive jet injector system, which works on the combustion platform uh, made by Diacel in Japan. And then there is phospholipid vesicle platforms. And interestingly, there is another candidate by SimVivo, which is in phase 1 clinical trial, which uses a bacterial vector for delivery, something like one used to do in the classical for the typhoid vaccine like Vivotif. So this is the developmental journey. We started the development of this vaccine when the sequence was published for the Wuhan isolate. And in February, we worked out on the sequence, got the sequence, the construct made outside India, and then took forward the fast track development of this candidate vaccine. The vaccine was found to be stable for a couple of months at 25 degrees Celsius. We found a very strong immune response in both animals and now in humans, was able to neutralize the wild type virus. There were no safety concerns. And the preclinical work and the phase 1 trials are already published. We have already submitted our phase 3 interim data for publication in a peer reviewed journal, and hopefully that should be out in a month or so. So some of the challenges which we faced at the onset of this program was there was no human DNA vaccine available, so we did not have any established platform. And specifically in India, we did not have any DNA platform at a commercial scale available. So we looked at first the vaccine design, and obviously there was a lot of risk because a lot of clinical trials had happened in the past using DNA vaccines, but none of them could move to the commercialization stage. But there was good evidence for some of the candidates which had gone into advanced clinical stages, specifically for SARS-CoV-1 and MERS coronavirus, where due to the cases of the disease going down, it did not move forward to a phase 3 clinical trial. And that helped us, gave us confidence on what could be the kind of strategy we can make in selection of the target gene, the sequence, the promoter, the signal sequence, and most importantly, what kind of a vector which has a much better safety profile, which we should be using for working on this candidate. Then selection of a cell substrate was very important because one needed to amplify the plasmid to produce the plasmid DNA. Subsequently, we started looking at different guidelines and what kind of analytical methods would be required and animal species would be required for evaluating the dose for this vaccine candidate. And this involved determining of both substrate and process related impurities, product purity, different isoforms. We also looked at what kind of supercoil content would be required or open circular content would be required for the plasmid DNA to provide effective immune response in small animals. We did not have any idea about what kind of a species we should be using for evaluating this immunogenic response. And we tried a number of animals in our animal house facility. And here we were lucky because Zydus has a ALAC accredited animal house. We have a huge animal facility, including a non-human primate facility, which provides us ample opportunity to evaluate different animal species to take this candidate forward. And once the process was defined, we were able to define critical process parameters and critical quality attributes for a vaccine candidate as we move forward. Again, the process development is something what many of you would know based on the Kaizen kits which are available within a conventional research lab. People do a simple lab scale purification for the plasmid DNA, and this we had to translate into a commercial scale process, a non-infringing process, starting from cell lysis and clarification, then concentration of the isolate, separating out the cell debris, taking it forward to chromatography, Again, there were a lot of articles or patents we refer to where multiple chromatographies were used, but we wanted to develop a process which is scalable, economical, because we were looking at a pandemic situation. We wanted to have the vaccine which could be given to the masses and affordable in nature. So again, we looked at a single chromatography, and again, the selection of the matrix was very critical at that stage, followed by ultrafiltration and finally sterile filtration. Another major aspect was looking at different formulations, and we tried number of formulations, looking at different buffers, ionic strengths, pH, stabilizers, excipients, polysaccharides, polyols, adjuvants. We even tried liposomal formulations and evaluated how the immune response looked in the animals. And finally, we found that probably the naked DNA was the best suited to take this forward, and that is what we put forward as a candidate. Again, the delivery device, we evaluated number of lactoporators, pyrodrive jet injectors, needle-free systems, and finally, we moved forward with a PharmaJet Tropis needle-free injection system, specifically related to the cost and use. It eliminated the needle stick injury and sharp disposal cost. It was a compact device that is easy to use. It was preferable for patients and vaccinators, doesn't cause pain and crying for patients. Compared to electroporation, which is supposed to be the best way for delivering the plasmid DNA, as one would have seen in research labs, we found that it was comparable in delivery of the plasmid DNA, but with much lower reactogenicity as seen in a electroporation device. Doesn't require any power source, which would be very important once you go into the field to the remotest part of our country. And obviously, it provided a higher immunogenic response in terms of both humoral as well as cell-mediated immune response. And most importantly was that this device had also received acceptance globally from different regulatory bodies, including WHO, FDA. It's a CE mark from Europe, DCGI, NVISA, etc., which gave us confidence of using this device for our vaccine candidate. Talking about the safety, which I think Dr. Arora mentioned briefly about this vaccine. So the vector which we used for our candidate is has been developed as per the FDA guidelines for consideration of plasmid DNA vaccines for infectious disease indication, has been demonstrated to be very safe in multiple clinical trials earlier. In a preclinical toxicity studies, and all these tests we were doing in a sequential manner. We never went in a parallel fashion and never skipped any stage of safety, because safety is something we were very we wanted to be very sure about any new candidate which we are working on, because I think safety would be the most important aspect before even the efficacy or effectiveness of the vaccine. In preclinical tox study, we did a repeat dose toxicity study. It was very well tolerated at 6 milligram dose in rats, which is 3 times of the current approved vaccine, which is 2 milligram a single dose in humans, and a 3 milligram dose in rabbits, which is 1.5 times of the human dose. And this was given by N+1 rule, which was 4 times the animals were vaccinated against the 3 times regimen. What is done in human, it was found to be very well tolerated in the animal species. We did a biodistribution study in rats, even though the guidelines say it is not required, but we still did these studies, and we found that the plasmid DNA cleared off within a couple of weeks post-injection. In a phase 1 clinical trial, all our volunteers were monitored for 24 hours in an ICU setting and subsequently for 1 week to evaluate complete safety profile before we moved into a phase 2 clinical trial. And now, as Dr. Sherville mentioned, that we have demonstrated safety and efficacy in over 28,000 subjects for the ZyCoV-D, and it has excellent safety profile for this vaccine. We also looked at the anti-nuclear antibody profiling. Over 23 panels of anti-nuclear antibodies were checked in over around 100 samples, clinical isolates, and we found, or the clinical samples from our clinical trials, and we found that none of these samples had any anti-nuclear antibodies, suggesting about the safety profile of this vaccine candidate. And recently, WHO has also published a guideline which is TRS 1028. They have upgraded their old guideline of 2013 or 14. And this also talks about the safety of different DNA vaccine candidates, and so does the US FDA guideline of 2007. This is just comparative data from a meta-analysis which we have done from a published paper, and looking at the different kind of reactogenicity in a local adverse event for different types of candidate vaccines. And as you would see that the DNA platform, it includes in fact the Inovio candidate also, and this is the ZyCoV-D platform. So you would see the DNA vaccine platform has the minimum level of reactogenicity compared to either an adeno or chimp adeno platform or an mRNA candidate or inactivated vaccines or subunit vaccines which are available, or the clinical data which has been published so far. Similarly, the case if one looks at the systemic adverse events, again you find that the DNA platform has the best safety profile followed by inactivated vaccines, whereas other platforms has a lot of side reactions reported. Another challenge which we faced was the setting up of infrastructure for manufacturing of a plasmid DNA vaccine, and that too during the pandemic time. And we were able to set up one of the largest plasmid DNA vaccine plant for manufacturing of the antigen in a period of around 10 months, and that was at the peak of the pandemic. And it houses one of the Asia's largest chromatography columns, one of the largest in the world cross-flow system. And all of these are imported equipments, and getting the engineers, getting the equipments, getting the installation done. I think a tremendous effort made by all our engineering, procurement department, and manufacturing teams to have this plant up and running. Similarly, we have also been able to now set up a syringe and adapter manufacturing facility for Pharmajet Tropis in India, and we have been partnering with the best of technology companies in the world to get these lines filled, where we would be able to make seven different components of the syringe, and they would be assembled in India, which will make this vaccine completely indigenous, including the device part being made in India now. Some of the unique attributes of the ZyCoV-D vaccine are, as I mentioned, it is thermostable. It is stable at room temperature for a couple of months at 25 degrees Celsius, which would really benefit in the distribution and handling of this vaccine. It does not show any impact on multiple freeze-thaw cycles. As you would know, that a lot of vaccines on freezing lose their potency, but our vaccine can undergo freeze-thaw cycles. So in case of transportation, the temperature drops sub-zero, there will be no impact on this vaccine candidate. And we have also done an open vial study where we find that the vaccine is usable up to 28 days after it is open. So for a multi-dose presentation, it would be a great plus when it is being used in the masses. As far as the delivery device is concerned, it's a needle-free device, so eliminates the risk or the fear which a lot of people have of needles, specifically in kids. I think it would be greatly beneficial for the delivery of the vaccine. It eliminates the needle stick injuries and significantly reduces the sharp waste management. As I mentioned earlier, it's a plug-and-play technology, and one can make new candidates rapidly. In fact, we already have vaccine candidates ready for alpha, beta, kappa, delta, delta plus, and lambda at a lab scale, and already being tested in animals and found to be immunogenic. So if required, one can always move to a new variant very rapidly. And we are working on a lot of digital support, which utilizes an anti-counterfeit measures in the packaging, so avoiding any fake vaccination campaigns. We are also supporting digital infrastructure, and it can be easily integrated and work with the government infrastructure of CoWIN and DevOps. So, and it can also be used in looking at the pharmacovigilance and AFI reporting for the vaccine candidate. So I think a lot of effort has gone, and finally I would like to acknowledge all the clinical volunteers, clinical investigators, regulatory agencies, and collaborators for continued support. Dr. Swaroop and her team has been like a pillar and supported us throughout both technically as well as funded the program for preclinical and clinical trials. Isha, the lab which is supported again by DBT in Pune, is a lab where we did all wild-type utilization assays. ICMR NIV supported us for a non-human primate challenge study, and PharmaJet Incorporated supported us with the device. With that, I'll just conclude my presentation. Thank you so much.
Thank you so much, Dr. Patel and Dr. Kapil. Thank you, Dr. Patel, for sharing the innovative research and the impressive portfolio at Zydus. And thank you, Dr. Patel, Dr. Metzel, for sharing the rigorous COVID vaccine development, PsychoVT, at Zydus. We look forward to more innovative technologies being developed. Now, moving on to our next speaker, um, the Coalition for Epidemic Preparedness Innovation, CEPI, is a global partnership launched in 2017 to develop vaccines to stop future pandemics. CEPI supports the development of vaccines and platform technologies against deadly diseases for which no licensed vaccines are currently available. I would now like to invite Dr. Rensky Hesling, Senior Scientist, CEPI, to share her thoughts on the development of DNA vaccines for other disease interventions. Over to you, Dr. Rensky.
Thank you very much for the introduction. Good morning, good afternoon, good evening, everyone. My name is Renske Hesseling, and I'm with CEPI, the Coalition for Epidemic Preparedness Innovations. And I will talk to you today about how we see DNA vaccines play a role in the response to other infectious diseases than just COVID. Next slide, please. So CEPI is a global partnership that was founded in 2017 in the wake of the Ebola epidemic, one of the previous large epidemics, when people realized that the world was not well prepared for the next outbreak. So CEPI was founded as this global coalition to create a world in which epidemics and pandemics are no longer a threat to humanity. And the tool we chose to do that is the development of vaccines, the acceleration of vaccine development, and also importantly, making these vaccines equitably accessible to the whole world. And I think what we've seen with COVID is that this second part of equitable access is also of key importance to fight pandemics. Next slide, please. So initially, CEPI focused on a core portfolio of 5 priority pathogens selected from the WHO priority pathogen list. These are MERS, Lassa, Nipah, chikungunya, and Rift Valley fever. And we have a portfolio of about 20 different vaccine candidates across these 5 disease areas. We also realized that the next pandemic may be caused by a new pathogen, which is why we invested in platform technologies to develop vaccines against disease X. So very easily adjustable platforms that could rapidly be deployed for any new pathogen. And of course, as we all know, that new pathogen turned out to be COVID-19. So building on our disease X portfolio, as well as our MERS portfolio, of course, another coronavirus, we could very rapidly build up a COVID-19 portfolio. And in fact, this is now a large part of our overall vaccine development. Next slide, please. So the efforts by CEPI and of course by many, many other people and companies and institutions have definitely paid off in that we have developed a vaccine and multiple vaccines against COVID-19 in record time. In the slide, you see the average time it took for, or the shortest time it took for various vaccines against various pathogens to be developed in the past. And you see that COVID is a record-breaking case. So we have made great advances in vaccine technology, really when we were challenged by this pandemic. Next slide, please. So despite this great success at CEPI, we think this is maybe not enough, or we can potentially do even better, which is why we set a very ambitious goal of developing the next vaccine in response to the next outbreak in 100 days. So this is threefold faster even than the fastest vaccine today. But we think this is necessary if you look at the graph at the number of deaths that had occurred by the time the first vaccine was licensed. This time, after 314 days, and how that can then be improved if we do it faster, that's really a tremendous impact on human life. And perhaps DNA vaccines, as one of the key rapid response technologies, can play a role in this aspirational goal. Next slide. So here is an overview, very similar to the one that Dr. Maitel showed, of DNA vaccines currently in development. I also added vaccine developments that are not just focused on COVID, but on other infectious diseases, and of course on oncology, since many DNA vaccine efforts were initially focused on oncology. And like Dr. Maitel, I also noticed that different delivery technologies and delivery routes are chosen by different companies. Electroporation and jet injection seem to be the most favorable, but there are other technologies there as well, such as skin suction, formulation technologies, adjuvants, oral and nasal administrations in different vectors. So there is still a lot to be explored here. Next slide. CEPI also recognized the promise of DNA technologies, and we partnered with Inovio in 2018, rather at the beginning of CEPI, to develop vaccines for Lassa and MERS. And in 2020, of course, COVID-19 was added to this partnership. All three vaccines are based on the Inovio platform of designing a DNA plasmid with an optimized antigen sequence, delivering this intradermally, and then following up with electroporation to deliver the plasmids to the nucleus. Some key achievements on the programs other than COVID is for INO 4500, which is the Lassa candidate, that this was the first Lassa fever vaccine candidate to enter human clinical trials. The first trial was successful, and it's currently in a phase 1b trial in Ghana, while INO 4700, the candidate for MERS, has entered phase 2 in the Middle East, following a promising phase 1 study in Korea. So on the next slide, I have some key data from these projects. Next slide, yes, thank you. So this is some key data from the Lassa program. What you see is NHP data on the bottom, the humoral responses on the left, and the cellular responses in the middle in a durability study. So you see quite high responses just after vaccination, but then they reduce over time to much reduced levels after 12 months. But then when these NHPs are challenged at 12 months with a lethal Lassa challenge, you see that they have 100% survival in both dose groups. So this was very promising and gave us great hope for the clinical trials. So the phase 1 data was also successful. This confirmed the broad cellular and humoral responses in humans. And currently, as I said, the program has moved on to phase 1b in Ghana. On the next slide, you will see some MERS results from the phase 1b trial in Korea. There were 3 groups. On the left, the humoral responses for the low dose, in the middle for the high dose, and on the right, there was a 2-dose group with a longer time interval between doses. You see a clear dose response. The high dose responds much better than the low dose, and perhaps also a positive effect of a longer dosing interval. So both these factors are now currently explored in the phase 2, occurring in the Middle East. Next year will be an exciting year for this Inovio-CEPI partnership, because the phase 2 MERS and the phase 1b Lassa data are expected in the first half of 2022. Next slide. So some advantages of the DNA vaccine platform have, of course, already been discussed. The thermostability has been mentioned, which is really the best platform perhaps for this aspect. It has a good safety profile and induces a broad immune response of both humoral and cellular responses. And the product is normally quite simple and highly characterizable, which makes technology transfer easier. Multivalent vaccines are possible. You can combine multiple antigens on different plasmids or even on the same plasmid. No anti-vector response is expected, so that boosting is possible. And of course, it is truly a rapid response platform that can be very easily adapted to a disease X And with Inovio, it was less than 3 months before they were in the clinic for COVID-19. Next slide. However, there are also challenges. The delivery method was already mentioned. Plasmid DNA needs to enter the nucleus to be effective. And different approaches are still being evaluated for that, which seem to have different pros and cons associated with them. A relatively high dose is required, especially as compared to mRNA vaccines, which can limit the capacity, and it can increase the costs, especially, of course, when very high numbers of doses are required, such as in the current pandemic. And then immunogenicity, a broad immune response is elicited, but the humoral responses are sometimes somewhat lower than with other platforms, and it is to be seen what effect it has for different disease indications. Next slide, please. So that brings me to my conclusions. So CEPI has recognized DNA technology already from the early days of CEPI as one of the promising rapid response technologies. It has key advantages in that it's easily adaptable, it has excellent thermostability, and induces a broad immune response. But there are challenges with delivery and manufacturing, and these will have to be overcome for DNA vaccines to reach their full potential. And on the next slide, I'd like to acknowledge, especially, of course, the Inovio team, whose data I have been allowed to present today. So if you can go to the next slide with acknowledgements, that is on the last. Yes, thank you very much for your attention.
Thank you very much, Dr. Hesling, for sharing the impressive innovative portfolio and the application of DNA-based platform for solutions for diseases beyond COVID. Thank you so much. So next we move on to the panel discussion. Today we have an amazing panel deliberating new directions in vaccine research. Our panelists need no introduction. We have Professor N.K. Arora, pediatrician and a public health expert, Executive Director in Pentrust. We have Professor V. Ravi, virologist and Nodal Officer for Genomic Confirmation of SARS-CoV-2, Government of Karnataka, India. Along with that, and then we have Professor Raghavan Vardarajan, a biophysicist and professor at the Indian Institute of Science, Bangalore. We have Dr. Kapil Mehta, President Vaccines and Diagnostics at Zydus Cadena. The session will be moderated by Dr. Hesling, Senior Scientist, CMC at CEPI. I would further like to request all the attendees to share their questions through the chat feature. Please mention if you would like to address the question to a specific panel member. With this, I would like to hand over to Dr. Retsky Hesling for taking the panel discussion forward. Over to you, Dr. Retsky.
Thank you very much. Yes, so we've talked today a lot about the first licensed DNA vaccine for COVID by Zydus Cadena, which is, of course, a great step forward for the field. So maybe I can start by asking Dr. Maital, what the key success factors were for succeeding with this vaccine, where many other DNA vaccines have struggled in the past.
Thank you, Dr. Resnick. I think, as I mentioned, there were a lot of factors. I think when we started, we were totally naive in the area, and we had a lot of things to address. And probably that helped us a lot because we were sure that it would do well in the human clinical trials, and that's what we got.
Thank you. And are you planning to use this platform for other disease indications now that you've been successful with COVID?
Yes, I think once now this platform has been established and we also have a manufacturing facility. In fact, we are not only trying to look at in-house development of some candidates, there has been a lot of discussion and a lot of interest which has happened from globally, where people have been working on different DNA vaccine candidates. And we are in discussion with few of them, and if everything goes well, probably you will hear more about new candidates.
That sounds very promising. So, Dr. Aurora, I believe you've been involved in reviewing the data for this vaccine as well. Do you have some words to add on why you think this vaccine has been successful?
So, one of the, I think, highlight or the issue which I would point out was that the company followed a copybook pathway. That whatever, because it was a new kind of a platform, and therefore no corners were cut. I think that was important. And doing preclinical small animal studies and then non-human primate studies using the next generation plasmid vector. And so I think a stepwise process was very important. And then another thing which has given confidence is that the recruitment occurred during the peak of Delta outbreak. That is the second. So most of the subjects were recruited around that time. Two. Number three, that I think this is, as very rightly said, was the largest vaccine trial and very meticulously managed. One of the attraction for, in fact, for the whole country has been that adolescents were included. So, and in fact, we made couple one or two presentations that tell us where else it has been used in children, because the data, the interim data analysis has very few immunogenicity data, and they are going to present us maybe next few weeks about the rest of the things. One issue I would like to flag here, and that is, I think, greater awareness among the vaccine experts and vaccinologists and technologists, because still a lot of people are afraid of DNA being injected into a human being, foreign DNA. And that has been a kind of, I would say, a certain degree of intellectual barrier, that people want more data, and that kind of concern is there whether there is integration taking place. It's good they have done anti-nuclear antibodies, which I don't think they were it was part of the protocol, but they have done it. And in fact, we have asked them to do it for pediatric patients also. But to me, it appears that some kind of a webinar like this is organized in a series manner, for which gives the fundamental of DNA vaccine development and how this will be a safe thing. In fact, when we were presented some of the phase 1 data of HIV vaccine, which is primarily done in children, that was quite comforting that it has been done, although not much progress was done. So one takeaway lesson, what our group thought, was that technical experts need to understand the basics of DNA vaccine development, and how many of the concerns have been taken care of as technology is. I'll stop at that.
Thank you very much for your views. So that is then one of the challenges that have to be overcome to educate people more on this technology. I was wondering, when we look at DNA vaccines in general, what improvements the panel thinks are required for DNA vaccines to have a more significant impact for the next outbreak? Maybe I can give the word to Dr. Ravi.
Ravi, you are mute.
Thank you for having me on the panel, Dr. Eslink, and let me congratulate the Zydus team, Dr. Kapil, Dr. Patel, and others for making the world's first DNA vaccine for human beings. What I look forward to in DNA vaccines in the future is the following: A, multi-epitope DNA vaccines. COVID provided a great opportunity for quickly developing a vaccine, and we know that spike was the major immunogen, but then there are diseases out there where you require multi-epitope, and we also look forward to multi-antigen formulations. I think these are important research questions that need to be addressed for a number of viruses. A number of viruses, as a virologist, I can say. But what I also would like to see is quickly adaptation of this technology for chronic illnesses. There are a whole lot of chronic illnesses with great disease burden out there, starting from our own scourge of tuberculosis to HIV to a number of other ones. And of course, ultimately, translating it into the space of oncology, where there is a lot of scope. So I think DNA vaccines with the Zydus-CoV vaccine has shown that it is possible, and these are some of the things that we look forward to. Of course, I echo what Professor Arora said, that the common perception even among professionals is that this gets integrated into the host genome. So we need to convince our own community first with solid data that there is no integration. And how are we going to do it is a question immediately that needs to be addressed. So I'll stop here. If there are any more questions, I look forward to it. Thank you.
Thank you. And Dr. Varadarajan, do you have any views on which challenges need to be overcome for DNA vaccines to have the biggest impact?
Well, actually, I had a few questions which I have posted. Maybe Dr. Kapil or Dr. Pankaj Patel can answer those.
Can you maybe read out a question for us?
Yeah, sure. So, firstly, is there an antibiotic marker on the plasmid? Is the PharmaJet single or multi-use? What is the anticipated cost per dose? And how can the integration probability be measured? Is it zero or very small?
So I'll just take your first question regarding the antibiotic marker. So we use kanamycin as an antibiotic marker for selection. Coming to the second question about the PharmaJet device, the device what we are using can give almost what the manufacturer claims is at least 20,000 shots with a single device which can be given. Obviously, the needle-free syringes have to be replaced after each shot, and that is what we have set up a manufacturing facility as I showed in my slide deck. Regarding cost, at this stage, I may not be able to share that in this forum. I think, and what was your last question, Varadarajan?
How can the integration probability be measured?
About the integration. So, integration, in fact, there have been a lot of studies where people have looked at the different organs and tissues and looked at the tissues where the integration could have happened. And as I showed you the two guidelines, so that has come and shown that there is no integration happening with the plasmid DNAs, the new generation DNAs which are constructed, because they don't have a mammalian site of origin. So there is no risk of expression in a mammalian system as well as integration. So that has been very convincingly demonstrated, and the regulatory guidelines clearly elucidate that.
Thank you. Does that answer your questions, Dr. Varadarajan?
Yeah, largely, but now, at least for example, is having an antibiotic resistance marker, and something that's going to be put into very large numbers of people, is that an issue or not?
I did not get your question, Raghavan. Your voice was breaking. Can you repeat?
So is having an antibiotic is having an antibiotic resistance marker an issue or not for something that's going to be made in large quantities and injected, into people, and could these could the marker be acquired by commensal bacteria and other things, for example?
So, Narpan, that the antibiotic marker which we are using is something which has been accepted globally for expression. And specifically, ampicillin is the one where there is a concern. And that is what is not accepted by regulators. But kanamycin is one of the safest antibiotic markers which is used, and that is what we have used in our country.
Okay, I didn't have any further questions.
Okay, thank you very much. I would like to touch upon the delivery issue. So it was mentioned several times that different delivery methodologies are still currently used. So does the panel feel like we should all focus on maybe the PharmaJet now that it's become successful? Or is there space for different delivery methodologies, or may differ per disease indication? Dr. Ravias, are you nodding? Do you have a view on this?
I think the PharmaJet has several advantages, and A, it is proven successful. B, there are several advantages with terms of biosafety, especially avoiding needle pricks, and C, it removes the fear, the psychological fear in people of looking at a needle. So in more than one ways, I feel the PharmaJet is one of the ways forward, and it's far more technically simpler than electroporation. So that's my understanding, but I am willing to learn from others. Over.
Are there other views in the panel?
It certainly looks like there are a lot of advantages, but I think immunogenicity is still not very high, at least from the data I've seen. So anything that can improve immunogenicity would be good. So I think there's still room for exploration.
So I would like to make 2 points here. One is I would compliment the company for backward integration in a sense that they would be manufacturing here in India. So because the need is going to be with every dose there is a need. So rather than being dependent, I think all DNA vaccine manufacturers, some kind of a model will have to be developed because depending on where and when and how it is used. So I think it's a good idea, and I would like to compliment them on that. The issue is that following on Varadarajan's point, how to boost the particularly the humoral response, because the T cell response is very good. The humoral response is something, although with whatever we have seen, it is not as bad as it is made out in the literature, and that is to look at the how the plasmids are carried over to the germs nearby lymph nodes, because you are injecting in the intradermal area. So, are some studies being planned or done to see how germinal cell stimulation occurs? Because RNA vaccine with very high humoral response, but works primarily by stimulating the germ cells there. So whether something of that kind, and since there are plenty of lymphatics, and the whether it is somehow carried out, the antigen, the protein which is developed after injecting uh, is carried out, and what happens to the nearby lymph nodes also. So basically trying to see how that stimulation can occur, because that may be a game changer of sorts, that we have both good T cell response as well as humoral response. I wonder whether somebody would like to respond to that.
So I'll just like to add about the humoral response for ZyCoV-D. What we have found is that there is an excellent neutralizing antibody response for the candidate vaccine. So probably the quality of the antibodies which are generated and much better affinity and avidity, and we see a very good neutralization. And in fact, when we had compared with the convalescent sera, the ratios was almost like threefold, equivalent to what Moderna and Pfizer had reported in their papers. So I won't say the humoral response is inferior. In fact, it is better than many of the other vaccines which are licensed. And obviously, the T cell response was excellent in the vaccine. Even as Dr. Arora mentioned about the clinical trial, which happened during the peak of the second wave in India. And in fact, we looked at a subset of subjects who had a breakthrough infection. 100% of those subjects had the Delta variant infection, which even though we were not looking at an efficacy against the Delta variant, but probably what we got was against the Delta variant because that was present in the community at that point of time. And I think this candidate, if you're looking at the next generation, I think this is just opening the door. Now, with the first vaccine getting approved for human, I'm sure there would be a lot more researchers, both in academia and industry, who would be getting involved in more DNA vaccine development. I'm sure there is a lot of scope in developing better plasmids. There is a lot of scope of developing better formulations or delivery devices. But I think this is just the start. I think this field will see a lot of revolution in coming years.
I would like to say, add one any additional point, and it's a challenge as well as a request that 1 crore dose a month is not sufficient part of many of the Indian cities. So I think the manufacturing capacity should be, there has to be a way where these processes are in a way that larger quantity of vaccines can be manufactured. I do not know anything, but I think that is the demand because tomorrow, whether this vaccine is used a primary vaccine or a booster vaccine or for children, we need huge amount. We have 44 crore children. And I was thinking if I have to give 44 crore children, it requires 1.3 billion doses from. And as far as Zydus is concerned, it will take me 10 years for that. So we need a faster vaccine manufacturing facility. Over.
Dr. Ravi?
Question to Dr. Kapil and Dr. Patel. Where are you spending a lot of resources and time in this entire development? Is it in the post-processing, your purification? That's where your yields are going down? If so, it's a very naive, stupid question. Can you take a synthetic route? Like the mRNA guys have taken a synthetic route. Is it there in future or it's not possible? Over.
So right now we are not losing much process. It is just the process which has been scaled up. It's a very simple process as I showed in the flow chart. Obviously, since the requirement of the dose is high, you're talking about milligram dose compared to mRNA, which is dosed at a microgram. So even if one is making, say, 1 crore doses, and similar amount of quantity in an mRNA would have been something like 1,000, a million, 1 billion or 1,000 crores. So I think that is where the challenge is in translating into the number of doses. But the process is quite simple. It only needs to be scaled up. And I'm sure now since, as I mentioned, this process, this candidate has been successful, there would be a lot of development happening in this field, and maybe more manufacturing facilities would be coming up in India and globally. In fact, there have been a lot of interest which we are seeing from a lot of partners, both in India and outside India, who are willing to either extend their existing facilities or set up facilities in partnership. So I think there is a lot of scope to move forward. And another advantage which Dr. Arora mentioned is that this vaccine can be used as a booster dose. And I think we may not be able to vaccinate all 1.3 billion selectively by our vaccine, but probably we can provide a booster dose to all the ones who have been vaccinated, because a lot of vaccines, because may not, because of anti-vector immune response, may not be suitable for further booster doses.
So I think if I can also add to what Kapil said and Dr. I think we understand the issue, obviously, in terms of the need of the hour, but I think we just started off, we reached a level where we could now scale up to this level. We know we can go at least 3 to 4 times more, and with partnerships internationally where they have established DNA platform manufacturing, we can do it. But I think everything requires a little bit of time. But as I said, this is not something that's over. The journey still continues, and I do believe that over a period of next 3 to 6 months, we would have been capacities which are at least respectable in terms of the requirement that will be there. But to scale this platform at such levels, we are using cassettes, chromatograms, which have never been used, not in India, but even in many parts of the world. Even the tangential flow filtration system. I mean, to just design that by the manufacturers was a task. So I think we're getting there, but I think we are looking at the long term, and we do believe that this will be an effective solution. As Kapil said, that we have seen very good long-term response to this vaccine, and we believe that that could potentially be a good move forward multi-variant vaccine in the future.
Yeah. I had a question about boosters. In lab studies, normally people use DNA as a prime and something else as a boost. But here, you would like to do it the other way around. So is the boosting capacity equivalent to the prime? Because it's a very good prime, I know that. But is it also a good boost? Do you have data for that?
So Rahul, we have not tested currently in a prime boost approach, obviously because we did not have the access to the other candidate vaccines to test in the lab as of now. But I think moving forward, that would definitely be one of the approaches which we would like to evaluate.
Evidence to rule out any genomic integration of the vaccine DNA. Over to you, Dr. Kapoor.
So I think, as I already mentioned, the plasmid which we have been using has been successfully demonstrated that it does not integrate. There is a wealth of literature available, and that is what even the regulators have shown, that it doesn't integrate in the human genome. So I think that, as Dr. Arora also mentioned, there is a misconception about integration of the DNA vaccines. Probably that was there a few years back or a decade back, but now with so much of literature available, I think it doesn't integrate at all.
I think what to Kapil's point also, both the US FDA and WHO have very clearly stated about the biodistribution and integration data. So the vector that we have used is of that. So it's already very well put in terms of literature and data that it is safe from that effect.
Thank you. We have another question from Mr. Tarun Sharma. In fact, 2 questions. So first is, can antibiotic marker removed in future DNA vaccines? And the second, will microneedle help in dose sparing?
So regarding the antibiotic marker, yes, definitely it can be looked into using different kind of other platforms for selection. But obviously, antibiotic is the easiest selection, because one would be looking at not propagating something which is not sensitive to the antibiotic. That is the first part of the question. The second thing about the microneedles, yes, there's a lot of studies which are happening globally where micropatches and other delivery devices are being used for DNA vaccines. And time will tell how the data emerges.
Thank you, Dr. Kapil. Over to Dr. Hesley.
Thank you. Thank you very much. Yes, I had another question, perhaps for Dr. Mattel. If, in addition to increasing your manufacturing capacity, are you also looking at dose bearing approaches, which could be another way to increase the dose capacity?
Yeah, definitely. I think that will lead to the second generation of the plasmid DNA vaccine, either in terms of adding some sort of a formulation which will help in dose sparing, or a delivery device which will reduce the requirement of the antigen. So yes, that is something which we have been thinking of right now, but obviously nothing concrete has been worked out so far. But we would be open to any ideas or suggestions if SEPI has. You have a lot of partners and collaborators.
Are there other suggestions from the panel on this topic, on the use of adjuvants or delivery technologies?
Question I would like to ask is, one is I was also going to ask about adjuvants, whether that would make a difference that after transcription, whatever protein is produced, it gets adjuvenated and better immunogenicity, etc. Something so that which joins the molecule at a step after DNA translation has taken place. The other is uh, the has somebody done comparative study on a gene gun or a jet injector or some of the other exploration processes, and whether that would make a difference in the total vaccine and the antigen delivery, the quantity of vaccine delivery. Are there those such kind of comparative studies? So rather than trying to not to get worried at this point about the cost issue, because that obviously can be brought in, that's technology of a different time. But has somebody done that comparative study? Because ultimately this plasmid is to be delivered to the nucleus of the cells. What is the most efficient way, and so that the antigen requirement goes down?
I just add before you respond, I just add a modification to Dr. Arora's question. Is there a non-invasive delivery mechanism? Like the RNA guys have used liposomes to deliver it into the cell where it fuses with the cell membrane without causing any damage. Like that, is there any thought process? Are there any advances?
So, in fact, Dr. Ravi, the current device what we are using is actually non-invasive. It's a jet injector and needle-free. In fact, mRNA vaccine uses a needle and syringe for injection. So, in true sense, it is a non-invasive injection which is given in case of DNA vaccine. Talking about the use of different adjuvants, see, the primary objective once you are giving a plasmid DNA is that it should enter into the nucleus. So then the transcription happen and then the translation happening in the cytoplasm to make the protein. So one has to select a adjuvant very carefully that it doesn't inhibit the cellular uptake. I think that is very important to be done. And as I mentioned, we tried number of adjuvants. We tried different sugars. There are a lot of literature talking about chitosans and different polyols which help in a better cellular uptake. But we found that each one of them had their own limitations in terms of the amount of antigen which could be loaded on that, the uptake of the antigen by the cells. I think each one of them had their own limitation, and we found that the naked DNA was doing the job very well. Obviously, since we were in a rush, I won't say we did a very exhaustive formulation studies at that point of time. We tried our best. I'm sure there are ways where further the delivery could be improved upon. And probably the next generation DNA vaccines would have that kind of platforms. And we are also looking into some of them ourselves.
Just another, to continue on that. With patches, there's a lot of variation because of skin type and thickness and hair and all that. Is that a factor with the PharmaJet, or that's not a factor?
No, that's not a factor in case of a PharmaJet. There is an extensive study done by the manufacturer where they have looked at different kind of skin types. And that is one of the points which we were in discussion with them at the time when we were evaluating different devices. Because obviously, in the wrinkled skin or in a young population, there might be differences in the uptake. And they had some very interesting data at that point of time, and they showed us that it works equally well. So they have engineered in a such a way that the DNA could enter into the dermis of the volunteer who has been vaccinated very well. But yes, in micropatches, that could be a challenge. And obviously, loading the antigen in a micropatch needle device also has its own challenges in terms of the content which could be delivered and how to dispose of these micro needles. But I'm sure as the technology is advancing, the new generation micropatches being used for measles vaccine and other vaccines, I am sure there would be solutions on the road of development.
Yeah, thank you.
Thank you very much. I believe we're running out of time for the panel session. Are there any closing remarks people would like to share with the audience? If not, then I think we covered all main important topics. So then I would like to give the word back to the organizers.
Thank you, Dr. Hesling, and all the esteemed panelists for an interesting discussion. And thank you, all the attendees, for the interactive session. I'm sure there are a lot of lessons learned. And as Dr. Kapil said, and I quote here, this has opened the door, and we have a long way ahead. Thank you again. From this, we now move on to the end of the session today. I would request Dr. Mukherjee, the Mission Director, GCI, for the closing remarks and a vote of thanks.
Thank
Over to
you,
you,
Shikha.
sir.
Thank you, Shikha. Indeed, a pleasure to propose the closing remarks and the vote of thanks today. And what a great discussion and what a great talk we had today from each one of the speakers today. And first and foremost, I think our Honorable PSA, Professor Vijay Raghavan, gave a very, great kickoff start to this webinar today, speaking on the India's landscape of past vaccines, present and the future vaccines, what India can work about, and what India can catalyze things moving faster together. So I think that was a great kickoff to this webinar, which really followed in a very rapid space thereafter. Then Professor Arora, I thank sir for joining us today, PSA sir. Professor Arora, stepped in and gave us the regulatory landscape of the vaccine program, how it was evaluated, what stringent measures was followed to do that, and I think that really showed the way how the things moved in. Then they came the major thrust by the Zydus Cadila team led by Dr. Shravan Patel and Kapil Mehta on the technology developed and how the vaccine played a role and what were the challenges they faced to take the vaccine to the next level, and the support they received and the how what will be the next stage now, what will be the next schedule. I think that was a great initial talk to make us all aware of what was the challenges the vaccine had. Then the global scenario was brought in by Dr. Anseke from CEPI. I thank her. I think she gave a global landscape. What are the other vaccines? What these platforms can be used for? That was really important for all of us to let know. Then the panel discussion was really enriching, and a lot of questions which were discussed today really brought in up, and a lot of questions which came from the YouTube, our viewers from the YouTube were really important. Still, there were a lot of questions pending, but as of the time, we need to really close. So I think each one, our speakers today and the panelists really enriched us, educated us, and gave us the way forward how the DNA technology can be used not only for this pandemic but for other vaccine, other infectious disease to follow. It was a great. I literally thank all of you, on behalf of Department of Biotechnology and BIRAC for joining us today. But I have to make a couple of more thank yous today. I think our MD, Dr. Alka Sharma, and Senior Advisor DBT for guidance and support for organizing today. Madam Dr. Renu Sarup, Secretary DBT and Chairperson, it was her idea to do this, educative webinar for all of us, not only for India but around the world, to know how India's first DNA vaccine was developed, what was the challenges, and how it was addressed, taking into the funding scenario, the regulatory, the science aspect, how now it will be absorbed into the immunization system. So thank you, madam, for joining us. Finally, Shikha, you yourself, Mithika, Dr. Mithika, Shikha, who have been really working along with this, my colleague Shubhra here, who has been supporting, and the IT team at BIRAC, who was really working along it. Thank you, sir. Thank you, everybody. Have a nice evening and good night. Thank you. Thank you. Over to you.
Thank you, sir. Thank you, everyone, again. May I request everyone to please turn on your cameras for a quick photograph. And if I'm not mistaken, is that Dr. Pankaj Patel with you, Dr. Shravan? Could he also come in the view? Thank you so much. Thank you, ma'am. Nitika, just let us know once the screenshot has been taken.
Yes, it's been taken.
Thank you.
Thank you. Thank you, everyone, again. Good evening.
Thank you.
Thank you. Bye.